A lot can be learned from this colorful chemical rainbow, but more on that later. Today we're going to talk about polymers. Polymers are the large biological molecules that build all sorts of living tissue, from plants to the simple connective tissue that holds our skin to our muscles. Long fibers are polymers, single units that repeat and repeat and repeat. So I'm going to do a model here with some hot glue, and it's going to answer the question, how do we fit all that DNA in such a tiny little nucleus? So I'm going to stick the little hot glue bead to another colored pencil here. I'm going to start pulling it apart, and what you start to see is longer and thinner, repeating. I've had students stretch this up to about 160 feet, I believe. So that's how you can fit 6 feet of DNA inside such a tiny nucleus. Polymers are everywhere. They're the spindle fibers of mitosis, they're the chromosomes of mitosis, the cell walls of the onion. And a lot of polymer science has to do with the water molecule. Let's start off with proteins. Proteins are a really good example of a repeating polymer. They're long polymers of amino acids. So an amino acid has an amino group, which is a nitrogen and 2 hydrogens, and then it's got its carbon backbone and a carboxyl group, carbon, oxygen, double bond, and hydroxyl. This is a variable group right here, and the variable group I just represented with a purple bead, but they're complex sugary molecules that give specific chemical properties to 20 different amino acids. All right, so I'm going to bond these 2 amino acids and make what's called a peptide bond through dehydration synthesis. Dehydration synthesis is the removal of a water molecule that creates an opening for a covalent bond. So by taking a hydroxyl and a hydrogen, one from each, now I've got a free space, and I'm going to bond together my carbon with my nitrogen. So the carboxyl bonds with the amino, and now I have a carbonyl and an amide, and away goes the water. There you go. 2 amino acids are now bonded together. We call that a peptide bond, where the amino group meets the carbonyl group. So now we have a peptide bond between 2 amino acids. Every amino acid has the amino group and the carboxyl group, so every amino acid can bond with a subsequent amino acid, and we can make long polymer chains of amino acids. We're going to shuffle this along now, and let's bring in another amino acid. I'll place that water there so you can remember that we kicked out of water. All right, so now I'm going to take the next carboxyl group and remove the hydroxyl, the oxygen and hydrogen, and then I need to take a hydrogen from the amino group, and we're going to keep that bond space ready. Here goes my dehydration synthesis, also known as a condensation reaction because water appears. And now I'm going to take the carbonyl and the amide, formerly carboxyl, formerly amino, and we'll bond them together again. There's another water molecule. You can remember how that happened. So this is a polymer, a repeating series of amino acid monomers. And here's my other carbonyl and amide, and carbonyl and amide, so we can keep track of where things used to be. The purple beads, remember, are variable groups. Those variable groups will have personalities that interact to give the characteristics of the protein, but for now what we're really thinking about is how we make a polymer between different monomers. We kick out of water through dehydration synthesis, also known as a condensation reaction, and with amino acids we call that a peptide bond. Now, what if I wanted to put those water molecules back in? If I put the water molecules back in, now I can break the same bonds, and that's called hydrolysis. Hydro, water, breaking, lysis. So hydrolysis is putting the carboxyl group back together and putting the amino group back together, and by putting a water in, I've just digested that protein into amino acid. So we can do this down the line, and we can break a protein into its residual amino acids through hydrolysis. Let's try that again. I'm going to break that bond. Now, who might assist something like this? It's probably got to be enzymes, right? Chemical reactions of this nature don't really happen that easily. So if we get an instigator in there to lower the activation energy, we can do this hydrolysis. So a lot of digestion, breaking down of polymers and the monomers requires some instigation by an enzyme, as does the building. I'm going to repeat this now. Speaking of building, we're going to use an enzyme, right? We're going to build a polymer again. Let's float that water out and let's put this back together. And there it is, peptide bond. Over here I still have a carboxyl. Over here I still have an amino. So both ends are still open for business. And there's my carbonyl, my amide. And this is two monomers put together as a peptide. And away goes the water. Here's a HEXOSE sugar monomer. This would be glucose or galactose. I'm not an expert on which direction the hydroxyl groups go and what makes what. So we're just going to say it's a HEXOSE sugar. It's a 6-carbon sugar in a ring. And you can see that there's sort of this hokey pokey. You put your hydroxyl group in, you put your hydroxyl group out, you put your hydroxyl group in, and you spin it all about. Well, when you've got the hydroxyl groups, you've got prime sites for these bondings through dehydration synthesis or condensation reactions. So it would be very easy to take a couple of hydroxyl groups, and we'll put those together here. And how should I go about this? It seems to be sort of a three-dimensional geometry problem, doesn't it? Well, if I took 2 hydroxyl groups off, I'd have hydrogen peroxide, right? We're trying to build a water molecule, H2O. So I'm going to take the hydroxyl off of 1 carbon and just a hydrogen off the other. If I take this hydrogen off and leave that oxygen has a space to bond covalently. And put our water molecule together, and now they've taken the hydroxyl off of the other carbon. Couldn't my carbon bond with that oxygen? So what's happening right here is actually what our insulin does. It's going to remove blood sugar by building these sort of polymers of sugar as glycogen and storing them in our muscles and our liver for later use. And so I could just keep this same process going. Right now I just have a little sucrose or something. Actually, this is like lactose. But it's this 2 carbons holding hands by the oxygen that gives us that beautiful covalent bond through dehydration synthesis, where you turn monomer sugars into a polymer. Remember, it's the kicking out of a water molecule in both amino acids and in sugars that allows us to build these long biological polymers. If I want to break it apart, let's say I want to release blood sugar back into the bloodstream, I'm going to use glucagon this time instead of insulin, and I'm going to put that water molecule right back in. Let's break that apart. We'll stick our hydrogen back on that oxygen and our hydroxyl back on our carbon. And so through hydrolysis, glucagon can restore blood sugar by breaking down our glycogen and releasing those individual glucose molecules back into the bloodstream. There we go. Through hydrolysis, I've now broken that bond, and I've got 2 sugar monomers. So if we expand on the idea of a disaccharide and keep on adding more links of sugar, we can build large polymers of sugar like glycogen in the liver, or starch for plants' roots, or cellulose for plant tissue. Iodine is a common biological stain, and we often just use it to make contrast in a microscopic slide, such as this nice view of onion cells with nuclei. But iodine is also useful as a chemical indicator of starch. When iodine meets starch, the reaction turns a beautiful midnight blue-black. We can use iodine to trace flow through a cell membrane into a starchy model cell so that diffusion and osmosis can be understood. In these potato cells, we see cell walls and little plastids called leucoplasts that look like little glass beads or sand grains. We know potatoes are starchy, that's why we eat them. The starch is going to be used by the potato to hibernate and store energy and ultimately send shoots towards sunlight so the plant can break above ground and start doing photosynthesis after a long hibernation. When we stain potato cells with iodine, we see that the starch is present, but only in the leucoplasts. Thus, iodine really helps us visualize the cell, form, and function at a chemical level. We can see those polymers. One of my favorite experiments is to use a protein polymer, an enzyme called amylase, to break down another polymer, a carbohydrate polymer, called starch, or amylose. Not like that. We are going to take the enzyme amylase from the saliva and put it with the starch, but we're going to do it outside the body in a test tube. And we're also going to use a control that doesn't have any enzyme in it at all. We'll test these with both iodine and Benedict solution. The enzyme amylase will instigate breaking the polymer starch amylose into its basic monomer sugars using hydrolysis. To the left, we see the reaction of amber-colored iodine turning midnight blue-black when reacting with the starch. Iodine is a great indicator of polymer carbohydrates, the polysaccharides. To the right, we see that iodine is revealing a negative test. It stays amber, so there must not be any polysaccharides in the test on the right. In the next set of chemical tests, we see Benedict solution, a blue copper sulfate indicator that will go through an oxidation-reduction reaction with simple monomer sugars. The test on the left is a negative test for monomers, and the test on the right is a positive test, as it is turned orange, yellow, or greenish. Back to the iodine. Both contain the same starch product, but the second test tube was only tested after my own saliva containing the enzyme amylase had worked on the starches for some time. My enzymes catalyze the hydrolysis of the starch. So the same number of sugars may exist, but they're no longer in polymer form. They are now simple monomers. We can verify this result using the Benedict. You can clearly see the starch in the negative Benedict test. Even though there are sugars present, they are polymers and don't react with the Benedict. On the right side, we see the results after the starch has been sitting in my saliva again. The test clearly shows the presence of monomer sugars. Interestingly, though not pictured here, a control of just my saliva produces both negative results for iodine and Benedict. No sugars are coming from my salivary glands. This just verifies that the experiment is actually measuring change in the starch as it goes from a polymer to a monomer through hydrolysis. Another favorite of mine is the ripening banana profiles seen here. As the banana moves from very green to ripe to what we might call overripe, we see the starch profile decreasing and decreasing more, while the monomer sugar profile increases and increases more. Some people like a green, bitter, starchy banana. Most people like a ripe banana. And some people crave those banana bread bananas as they're incredibly sweet, as so much of the polymer carbohydrate has now been broken off to single monomer sugars through hydrolysis. Again, this experiment was done with iodine, indicating starch that we could see decreasing, and Benedict solution, indicating sugar that we can see increasing. When you take a walk in the woods and you look around and you see all the trees and the plants, the birds and everything, feathers and hair, all of nature's growth is basically a synthesis reaction process. A plus B yields AB. So the compounds from food are assembled into those large macromolecules that you see growing out there in nature. And it's important to realize that energy is actually stored in the molecular bonds. When we head on into the garden and uproot a carrot and eat it, or destroy a potato, what we're doing is we're digesting those macromolecules and we're converting them into smaller molecules. A decomposition reaction, that's where AB becomes A plus B. And very often that is going to happen through hydrolysis. So let's review. Large macromolecules in nature are built through dehydration synthesis, or condensation reactions, where a water molecule is kicked out and smaller monomers are joined together in covalent bonds that become larger and larger molecules. By contrast, hydrolysis puts water back in and breaks apart polymers into their original monomers. The transfer of energy through living things is often accomplished through manipulations to chemistry. Understanding condensation reactions and hydrolysis will really help you wrap your head around the way that matter is involved with energy transfer. Remember, biology is a dance. Nothing stays the same. The dynamics of nature are reflected both in the changes of matter and the changes of energy. Why do we need to understand polymer science? Well, polymers are everywhere.